Moving on from the membrane more to the internal components of the cell now. The different cell organelles all work together to make the cell what it is basically. So let's take a look at some of these organelles inside of here. Before we can even get into the organelles though, gotta look at what's around it, all the parts and pieces, the cytoplasm. Cytoplasm is going to be the generalized idea of everything inside the cell. Now, that is going to be not including the nucleus, but everything between the nucleus and the membrane. The idea is all the organelles, all the liquid, all the ions, all the particles, all those are part of the cytoplasm. So from the nucleus to the membrane is what we're looking at. Now, the mostly liquid portion of that is going to be the cytosol. The cytosol is not including the actual organelles. This one's primarily water-based at this point. So cytosol, the water-based liquid inside the cell, while the cytoplasm is going to be everything from the nucleus out to the actual plasma membrane. So we'll start with the ER, the endoplasmic reticulum. Now, this actually comes in two different varieties. You have a smooth endoplasmic reticulum, as well as a rough endoplasmic reticulum. The rough endoplasmic reticulum, as you can see here, has all these dots around it. Now, these dots are actually ribosomes. Ribosomes are going to basically be your protein synthesis, so making proteins. There's both membrane bound, which we see here, and then also free ribosomes just floating around. But these membrane-bound ribosomes are what make it the rough endoplasmic reticulum. So if they're producing proteins, the rough endoplasmic reticulum really is protein production. The smooth endoplasmic reticulum doesn't have any ribosomes, hence why it's called smooth. But now it has its main purposes are going to be lipid production. So when we start looking at the smooth ER, we're looking at a lot of phospholipid production. Remember, phospholipids make up the cell membrane. So while they're both endoplasmic reticulum, they have two different purposes. While the rough is more protein, the smooth is going to be more of the lipid production. Golgi apparatus. Now, the Golgi apparatus can be also known as the Golgi complex sometimes. But this Golgi apparatus is a series of flat kind of layers together, sometimes referred to as a cisternae. Cisternae is just flat membrane layers. But as you can see here, it receives substances from the ER, refines and packages them. Pretty much think of this as a FedEx or UPS store. The idea is you can bring things in, like the ER sends materials there, then it gets packaged up, all sent out, and then shipped off. The Golgi apparatus is a packaging and shipping center. It's going to receive items from your endoplasmic reticulum, package them up into small membrane-bound vacuoles, and then send them on off. Those can stay within the cell, or they can go through exocytosis and release the contents outside of the cell to go other places in the body. But the Golgi apparatus is packaging and shipping. Mitochondria. Now, the mitochondria itself is going to be a little unique in the fact that it has a double membrane system, not a bilayer. I'm thinking two distinct different membranes. Of this double membrane, the inner membrane, so the one that's on the inside, so you have the outer membrane, which is right here, then you have the inner membrane tracing like in here. The idea is that inner membrane is going to have enzymes that help to break down food particles. Those food particles are how it produces energy. The mitochondria is the powerhouse of the cell. It's what creates a lot of your ATP, a lot of your energy inside your cells. It's where you find the citric acid cycle, as well as the electron transport chain. Now, those will be parts of how you're producing ATP in the cell. Metabolism contain their own DNA, RNA, some other things. Well, metabolism, we know, are metabolic reactions, so it's just all catabolic and anabolic reactions, which means that there's a lot of different energy production as well as some energy consuming reactions occurring inside here. But overall, it's net producing for ATP. And then the idea of containing its own DNA and RNA. Why it's unique is it's not necessarily the exact same DNA the cell has. It has its own set of DNA that helps to code and produce its own systems. So the mitochondria has its own little kind of center here that center helps control itself. But overall, for an anatomy and physiology standpoint, we're really looking at mitochondria based on the citric acid cycle and its ATP production. That's the powerhouse. That's where you get your ATP from. So looking at mitochondria. Lysosomes, peroxisomes. Now, I pulled the big image back up of the whole cell because when you start thinking of these ones, you can have, for example, a lysosome up here. These lysosomes can be found throughout the entire cell. There's one down here, there's one over here. The idea here is lysosomes are containing enzymes. Now, these enzymes usually can help with phagocytotic aspects because they're going to break down whatever is put into that vesicle. So a lot of times lysosomes helps with phagocytosis, removing dead and debris, just getting rid of things. While peroxisomes, they also contain enzymes, but a lot of times peroxisomes are focusing more on lipid metabolism, so breaking down lipids, as well as chemical regulation. What I mean by that is, think about free radicals. Peroxisomes can absorb those and get rid of those free radicals. So the peroxisomes, the lysosomes, both have enzymes for digestion, but they have a little different purposes, a little different functionality. From the lysosomes, more phagocytosis, peroxisomes are dealing a little more with the lipid metabolism and breaking down free radicals and other chemicals that need to be removed. Centrioles. Now, centrioles, you can see, are these triplets of structures. As you go around, all these individual triplets make up this ring-like structure. The actual centriole here can help with the cell reproduction. So if we go back a slide for a second here, you can see here are the centrioles. You can notice the centrioles have these lines kind of radiating out. The reason being, centrioles are used in mitosis. Go next. So as they're used in mitosis, they'll separate out and create the two opposite ends of the dividing cell, kind of pulling the pieces apart, helping to create this framework that allows the organelles and then chromosomes and everything to separate out to the two sides. So our centrioles, helping with our cell reproduction, think mitosis and meiosis. But we continue on that with these cytoskeleton components. Cytoskeleton, think the framework, the structural building pieces inside your cells. You have the first one here, the microtubules. Now, microtubules give kind of like a structural outline to how some of the certain different organelles can be held in place. You then also have the next one, the microfilaments. These microfilaments, which there will be some cases of myofilaments in the muscles, but these microfilaments are going to be structural units that are used in future parts that we'll talk about. Like I mentioned, the muscles are filled with microfilaments. You can see this one says actin subunit. That's one of the two main components that are making up your muscle cells. And then the last one are these intermediate filaments. You can kind of see how they're coiled tightly together, all this keratin, giving more structural support again inside your cells. So while the cells might be filled with organelles and liquid, there still is going to be structural components helping out to keep them the proper shape and make sure things stay in place. But there still are a few specialized pieces to mention. The term microvilli, micro meaning small, villi is a protrusion. Microvilli are very small protrusions on the cell surface. If you're looking at the diagram here, We start with the intestines. From there, it's going to move over, and each one of these is a single villi. But we still have to get even smaller, because the actual microvilli are just these little pieces on the very surface of each individual cell. So those microvilli are those tiny little pieces just along the cell surface over here. So it really isn't even visible until you look at the microscopic cell level. The whole purpose? Increasing surface area. You'll find the microvilli in the intestines here, for example, because they're increasing the surface area to give more surface area for absorption of nutrients and liquids out of your intestines. So you have increased surface area, microvilli. Cilia. Cilia are going to be these hair-like projections you see on the surface of these cells. Those little hair-like projections can help to propel substances like mucus along the cell membrane. Now, this particular example is coming out of your trachea. That's your windpipe, trachea. It's going to help the actual cilia here help to move the mucus along the trachea. So some cells have some specialized features. From microvilli to the cilia to maybe for a flagella. Think of a sperm, the tail, the flagella, helps propel that cell, move that cell along. So all these different organelles and special structures and components, they're all helping to make our cells what they are, helping our cells to perform the tasks they need to do inside of our bodies. Now, are there more parts and pieces? Sure, there's more details and pieces, but this is a good overview to let us figure out how these cells are functioning as we move forward into future chapters.